Hello, distinguished professors, scientists, researchers, students, and other delegates. Today, myself, Dr. Kaushik Biswas, I am going to presents my research topic on herbicide tolerance in jute and wheat, current research perspective. I am from Department of Biotechnology as a postdoctoral fellow, Vishwas University, West Bengal. So, the points to be discussed is what is herbicide tolerance, why herbicide tolerance is a global burning issues in the introduction section, and as well as why glyphosate tolerance is very important concerns. And next, in the research case studies, I have used jute as well as a major crops, staple particular fiber crops, and as well as wheat as a staple crops, and the future perspective of HTGMC. So we can see here in the introductions of herbicide tolerance. Herbicide tolerance basically given in the definitions, where you can see the herbicide tolerance in the main plant should be up to level where the particular dose can be lethal for the weeds. This is the very simple definitions. So normal way weed can be controlled by crop rotations, crop cover crops use, tillage practices, and crop variety, improved crop variety, as well as some biological controls like use of some insects and some fungus to kill that particular weeds. The chemical control is concerned with the most popular approach where it is very efficient. So if we manage this particular chemical control by improve the herbicide tolerance in the main plants, then you can easily increase the crop yields up to a certain level. So we can see here crop yields losses at global level and national level. We can see most of the crops, major staple crops all over the European country and USA, corn, soybean, edible beans, sunflowers, it is mostly 50 to 30% level of infestation due to weeds. And in case of India, rice and millet crops has been more than 30% and 40% of the yield loss due to weeds. Where we can see the use of glyphosate. Glyphosate is very efficient and very good herbicide that is globally practiced in different managements of weeds. So in this particular websites ISAAA, where I have collected this information, that a number of 298 events that are only related to modification of EPSPS gene, that particular gene where the glyphosate is inhibited, glyphosate is inhibited, and this particular EPSPS gene is important to produce such very important amino acids like tyrosine, tryptophan, and phenylalanine. So if in this way, if we control this modifications of EPSPS, we can easily manage the use of glyphosates. So next one, the different approaches used till date in transgenic events, where detoxification of glyphosate is very important. The first most important and most used practices where first one is Orthobacter Entropy. From this particular bacteria, GOX gene has been isolated and it particularly catalyze that carbon nitrogen bonds and make into non-toxic residues. Here it is shown the particular AMP groups, and these particular groups has been differentiated from each other. Next one, cleavage of carbon phosphate bonding. Here you can see this is the phosphate bond. If you release that phosphate bond from remaining sarcosinate group, then again it will be very effective. Another gene GO from E. coli is also use phosphorus as a source of their energy sources. So in this way, again glyphosate can be inhibited. EPSPS second particular strategy is EPSPS insensitive to glyphosate. That is also very important. And today I am going to represent this particular part. And glyphosate acetyltransferase, that is another gene that can be initiates particular EPSPS gene. So there are two different methods. I have used two different techniques in case of jutes and wheat, where EPSPS standardization has been done in case of jute regeneration has been done in case of jute, and in case of wheat we have used in-planter transformation methods, and it is published. So in case of jute, we can see that particular explant of shoot, apical meristem, and cotyledon petal has been used. The regenerates are giving very good response. In the next stage, we have transferred to that particular regenerants to shoot growth media, and the variable responses are also shown in this figure. And jute plantlets, when it is the shoot length is bigger, we have transferred to particular root initiation media, and after that we have transferred to soil rights with half ms for hardening and other purpose. In this way we have made ready particular jute plants for transgenic events. So the next one, in case of wheat, we have used particular steps where picambia 1301 has been used as a main transgenic constructs. Here you can see this particular thing has been used as a selectable marker HPT2, CP4 PSPS, that is modified, codon modified, and as well as GAS gene as for selectable markers. Scorable markers. Now you can see this particular seeds has been used at the very early stages, in the 2 days old seeds, where we have injected the agro inoculation with particular gene construct, and after that we have transformed into the plants until they get a particular stage, and at the T1 of that particular events we have used these particular sites, and finally we got the transgenic plants and transgenic events in the T2 generations, and we have used for further studies for molecular analysis. So future perspective, this is very important. Oxalic herbicide stacked with glyphosate or glufosinate in the next few years can be used. CRISPR-Cas9 based technology and RNA interference also get popular, which can be utilized in case of herbicide tolerance in the next few years. And the most serious concern about the shift of wheat species composition. So wheat is also trying to develop its resistance. So that is also we have to be clear. So in this way I can finish my presentation. So this is some reference I have used in my research studies and in presentation also. This is my own publications here, and some of the important publications has been given. Thank you very much.